Assalamu alaikum. Welcome to the next class. Related to the chapter part 4 of the course about the Mendelism. Mendelism basically covers all the aspects of the experiments conducted by Gregor Mendel and then the subsequent Mendel laws. We can start with human example. Uh, you might have listened about the black urine. Technically, it is called alkapton urea, and mostly it is observed in a family among the first cousins. So, a figure shown on the left side of the slide, you can see a normal urine in A and a black urine B, which is because of this disease, uh, alkapton urea. On the right side of the slide, you can see a complete biochemical pathway through which the phenylalanine is converted eventually into the malloyl acid. Now, during this path, just before the final product, you can see an enzyme um, that is called the homogeny gentisate 1,2 dioxygenase is not working properly, and because of that, homogenistic acid is not converted into the mallyl acetoacetic acid, and that results into the black phenotype in the urine. Uh, now, this black urine disease have some other symptoms as well. Those are clinical symptoms. The urine color is black. Then there is organosis, some sort of rashes around the mouth. Then the tendonitis, that is the problem of the tendons. Then the kidney stones, prostate stones, and then the kyphosis, or that's the bunch bag. So these are the common issues or clinical symptoms related to this disease. Now, why I am quoting this example is just to let you know the genetic importance or part of the genetics being played in the inheritance of this disease. So this disease is basically inhuman because of the defected copy present on the chromosome 3. So this chromosome is not that of the sex chromosome. So basically this is not the sex related disease. You can see a yellow arrow that indicates the position of the band on which a defected copy could be present and result into the disease. So you can see two parents which are basically carrier or the heterozygotes for this uh, gene. Now the affected individual or the color for the defected copy is the blue, while the normal copy is indicated in dark orange color. So both of the parents, they are basically carrier. So if they get married, what would happen? So there is probability that one fourth of the child, each fourth child that would be affected, because that child is going to get a defected copy from both of the parents. Similarly, one fourth or the every fourth child is expected to be unaffected or the normal child, because that is getting the correct copy from both of the parents. And half of the offsprings, they would be carrier for the disease just like the parents, because they would be the heterozygous for the genes. So this is basically autosomal disease. So there would be no effect of the sex. Indifferent to sex, the offspring are expected to be uh, affected or not affected. Now, this can lead us to the experiments conducted by the Mendel, because here you can see the classical ratio 1 to 1. So actually 1 is normal, affected, 2 is not affected but carrier, and then again 1 is the normal. So Mendel, he was the father of the genetics. He did some classical work on the genetics. We have already studied the history, and you are advised to go through again the history chapter and the contribution of the Mendel. Why Mendel was so successful? Because he opted for the pea plants. They are easy to raise. They have short generations, so results could be achieved in short span of time. And he produced many offspring, and he had large number of varieties. And then finally, the most important component of his success was the way he conducted the experiment, or his experimental or scientific approach on the subject. So characters that were studied by the Bendels included: number one, seed endosperm color, it could be yellow or green. Then the seed shape, it could be round or wrinkled. Then the seed core color, it could be gray or white. Then the flower position on the plants, axial or the terminal position. Then the stem length, whether it is short plant or it's the tall plant. For pod, you study the pod color, yellow or green. Then the pod shape, inflated or constricted. So these six characters were studied by the Mendel to establish his laws. Now there is some genetic terminology which we must know, as inferred from the studies of the Mendel. Gene, a genetic factor, or now it could be called the region of the DNA, a strand of the DNA that helps to determine a character. That's called the gene. Then the allele, one or two more alternate forms of a gene. So normally in dizygote, in the dizygote, and in the animals which have, or the plants, animals or human which have the two copies of the gene, so it would have two alternate forms. But in case of plants, there could be more than two alternate forms. So locus, a specific place on chromosome occupied by an allele. At this point, I would suggest you to revise the practical part related to the chromosomal structure, genes, and this sort of stuff. Then the genotype is basically a set of alleles that an individual possesses. So this is the combination of both of the alleles in case of human or animals. Heterozygotes, an individual possessing two different alleles at a locus. So that's the heterozygotes. Homozygotes, definitely having two same alleles on a locus. that is called the homozygotes. So homozygotes either could be dominant or could be recessive. Then the phenotype, the appearance of the or the manifestation of a character. So basically this is the expressed form of the gene. Characters or characteristics, any attribute or feature, that is the basically phenotype description. And just in previous examples are the traits studied by the Mendel. It could be the color of the pod, shape of the seeds, and likewise. So I'll try to explain the Mendel work with the monohybrid cross. So we use allele symbol in genetic description or writing the genetic studies. Allele symbols, capital means that is the dominant allele. Here in this case, for instance, it is capital A, so it's the dominant. Then the small alphabets letter that indicates the recessive. Here is a mistake. The recessive is also indicated by the capital. It's not the capital, it should be the small a. Then the genotype, two alphabets or the letter. The combination of all the allelic forms. So normally there are two. So in the possibly it could be both of the alleles could be dominant, it could be heterozygous, and both could be the recessive. Now P, capital P, that stands for the parents. F1, that stands for the first filial generation. F2, that is the second filial generation. So monohybrid example of the Mendel cross, the purple flower, that is having the genotype of capital P, that means both of the alleles they are dominant and the color would always be purple. And if these plants they are intercrosses, we are always going to get the purple color of the flowers. Similarly, white flower, and they have the recessive gene, that is small p, both of the alleles they are recessive, and as long as they are intercrossed, the result would be the white flower. So these are basically the parents. So when these parents are crossed, so what we get the F1 generation, and if when in F1 we will get all of the parents offspring heterozygotes. So as the P is completely dominant on the small p, so we will get the only one phenotype, that is the purple flower, for all of the offsprings. So genotype is the same, capital P, small p for all of them. Well, when this F1 is intercrossed, heterozygotes purple is crossed with the heterozygotes purple, that's the intercross of the F1. So then we get the F2, and in case of F2 you can see the phenotype, there are three purple and one white flower. So genotypes, there are two heterozygotes, one dominant and one complete recessive. So phenotype ratio we get 3 to 1, and genotype ratio we get 1 to 1. Okay, so this study can also be repeated with the Punnett scale, in which it's much easy to demonstrate and much easy to understand each of the individual alleles. So we write male and female. So normally the columns indicate the male and the rows indicate the female. Now here we are using, again repeating that experiment, a completely dominant male with the capital P purple flower, and it will produce two alleles, and each one of them would be dominant. Similarly, a completely recessive female having both of the small p's and having white flower, and it will produce small p as allele. So here when capital P and small p combine, it would be heterozygous, and it will be true for all of the offsprings. So they are all of them purple in color and heterozygous in term of genotype. Now, we intercross this F1 generation, and this is basically heterozygotes, and they will produce two type of alleles, capital P and small p. Similarly, the female will produce two type of alleles, capital P and small p. And when they are intercrossed, we will get capital P, capital D together, and it would be dominant. Both of the alleles, the color would be purple. Similarly, capital P, small p, result is heterozygous. Capital P, small p, the result is heterozygous. And one small p, the result is homozygous recessive, and that is basically white in color. So this was all about the monohybrid crosses which were done by the Mendel during his experimental studies, and how these could be applied in the human or the animal life. I have already shown you with example at the start of the lecture. So please watch the lecture carefully and try to understand and grab the concept of this monohybrid cross, and stay home, stay safe, have a wonderful time. Thank you very much. Allah Hafiz.